Welcome to this lecture on the fundamentals of genetics. Today, we are going to explore one of the most fascinating and revolutionary discoveries in modern biology, the ACDS elements in maize. If you have ever looked at a decorative cob of Indian corn and wondered how its kernels get those beautiful, unpredictable purple spots and streaks, you are about to find out. This topic will completely change how you think about DNA, taking it from a static instruction manual to a dynamic, moving machine. To start our journey, imagine you are reading a beautifully written recipe book, but suddenly a whole paragraph magically unglues itself, jumps a few pages ahead, and pastes itself right in the middle of a cake recipe. The recipe is now completely changed. In the world of genetics, this exact phenomenon happens inside the DNA of living organisms. We call these jumping paragraphs transposable elements, or jumping genes. The most famous example was discovered in colorful Indian corn, or maize, by the brilliant scientist Barbara McClintock. Those beautiful purple spots you see on decorative corn are the direct result of these genes moving around during the kernel's growth. Now that we understand what jumping genes are, let us look at how the specific ACDS system works. This system is a classic example of class 2 transposable elements, which physically cut themselves out of one location and insert themselves into another. The system has two main players. First is the activator, or AC element. It is autonomous because it produces an enzyme called transposase, which does the actual cutting and pasting. The second player is the dissociation, or DS element. It is essentially a broken version of AC and cannot produce its own transposase. Therefore, DS is completely stuck unless a functional AC element is also present in the exact same cell to provide the enzyme. Before we move on to the mathematics, let us see this system with a concrete visual example. On screen, you are looking at three different maize kernels. On the far left, we have the wild-type kernel, which is solid purple because its color gene is fully functional. In the middle, we see a solid yellow mutant kernel. Here, a DS element has jumped into the color gene, breaking it. And because there is no AC element present, it is permanently stuck. Finally, on the right, we have the variegated, or spotted kernel. Here, the DS element is in the color gene, but an AC element is also present elsewhere in the cell. The AC element produces transposase, allowing the DS element to jump out in certain cells during development, restoring the purple color in those specific patches. Having seen how these elements create beautiful patterns, we must ask why this discovery is so fundamentally important to biology. Understanding the ACDS system completely shattered the old scientific belief that genomes were static, unchanging instruction manuals. Instead, it proved that DNA is highly dynamic, constantly shifting and rearranging itself. This movement is a major driver of genetic variation and evolution. Today, plant geneticists use this exact system as a powerful molecular tool. By intentionally letting these genes jump and disrupt other genes, scientists can figure out what specific genes do, helping to breed crops with better drought resistance or disease immunity. This pioneering work was so revolutionary that it earned Barbara McClintock the Nobel Prize. While genetics is heavily biological, we can mathematically model the behavior of these jumping genes. Barbara McClintock noticed a fascinating quantitative relationship. The number of AC elements directly affects the physical size of the spots on the corn kernels. This brings us to our first formula. The average spot size, represented by S, is equal to a biological constant, K, divided by the AC dosage, represented by D sub AC. The constant K depends on the specific genetic background and cell division rate. The dosage is simply the exact number of activator elements in the genome. This formula tells us that spot size is inversely proportional to the number of AC elements. If the dosage increases, the transposition happens later in development, which means the restored cells have less time to divide, resulting in smaller spots. Now that we have our formula, let us apply it to a real laboratory scenario. A geneticist is studying two different maize kernels grown under the exact same conditions. The biological constant for this strain is 120 arbitrary units. Kernel A has exactly one copy of the AIC element. We're asked to find its expected average spot size. We start by taking our inverse relationship formula and substituting the known values. We plug in 120 for the constant K and 1 for the dosage. By performing this simple division, we isolate the variable for our first test subject. The final answer shows that the average spot size for kernel A is 120 arbitrary units. This relatively large number indicates that the jumping event happened early, allowing a large patch of purple tissue to grow. Having calculated the spot size for our first kernel, let us see how an increased dosage changes the outcome for kernel B. We keep the exact same biological constant of 120 because both kernels belong to the same strain. However, kernel B has exactly 3 copies of the AC element, so our denominator becomes 3. We substitute these values into our formula to directly compare the effect of having 3 times as many activator elements. Dividing 120 by 3 gives us our final answer. The average spot size for kernel B is only 40 arbitrary units. We can clearly see that kernel B has a significantly smaller spot size compared to kernel A. So we have successfully calculated the spot sizes for both kernels. This brings us to a formal proof of Barbara McClintock's biological observation. In our first step, we lay out our results. Kernel A yielded a size of 120, while kernel B yielded 40. The mathematical reasoning is clear. As the dosage increased from 1 to 3, the resulting spot size decreased significantly. This mathematically proves the inverse relationship. But what is the intuition behind why this works? Physically, a higher dosage of AC elements actually delays the jumping action until much later in the kernel's growth cycle. Because the jump happens later, the color-producing cells have much less time to divide and multiply. This perfectly explains why we see much smaller, finer speckles of color when more activator elements are present. Now that we understand how dosage affects timing, let us look at the actual probability of a jump occurring in a single cell division cycle. The probability of the DS element excising, or jumping out, is directly proportional to the concentration of the transposase enzyme. The formula governing this is P equals C times the concentration of T. Here, P is the probability of the gene being restored. The symbol C represents the excision constant, which tells us the baseline likelihood per unit of enzyme. Finally, T in brackets represents the actual concentration of the transposase enzyme in the cell. If the enzyme concentration increases, the probability of the mutation being reversed increases proportionally, leading to more frequent genetic changes in the population. Before we conclude, let us see this probability formula in action with a concrete example. In a laboratory experiment, we have a population of maize cells where a DS element has deactivated a vital gene. We are given an excision constant of 0.05 and a measured transposase concentration of 4 units. We are asked to find the exact probability that the gene will be restored in a given cycle. We choose our linear probability formula because it perfectly models this biological interaction. We carefully substitute 0.05 for C and 4 for the concentration. Multiplying these together gives us our final answer of 0.20. Physically, this translates to a 20% chance of the DS element excising, meaning about 1/5 of the cells will successfully restore the vital gene. We have covered a lot of ground today, so let us review the most important ideas. First, the ACDS system in Maize is a brilliant demonstration that genomes are not static, but flexible and capable of internal movement. Second, remember the roles. The activator provides the necessary machinery for movement, while the dissociation element is a broken passenger that can only move when the activator is driving. Third, we mathematically proved that the physical size of the color spots is inversely proportional to the dosage of AC elements. Finally, the concentration of the transposase enzyme directly dictates how frequently these genetic mutations occur. Understanding this system unlocks the fundamental mechanics of evolution across all living species. Great job following along with these complex genetic concepts.